Have you ever wondered if bacteria make distinctive sounds? If we could listen to bacteria, we would be able to know whether they are alive or not. When bacteria are killed using an antibiotic, those sounds would stop, unless of course the bacteria are resistant to the antibiotic. This is exactly what a team of researchers from TU Delft now have managed to do. They captured low-level noise of a single bacterium using graphene. Now their research is published in the journal Nature Nanotechnology. The research team was originally looking into the fundamentals of the mechanics of graphene. But at a certain point they wondered what would happen if this extremely sensitive material comes into contact with a single biological object. Graphene is a form of carbon consisting of a single layer of atoms and is also known as the wonder material. It is very strong with nice electrical and mechanical properties, and it is also extremely sensitive to external forces. The research team ran their first experiments with E. coli bacteria. The researchers says what they saw was striking. When a single bacterium adheres to the surface of a graphene drum, it generates random oscillations with amplitudes as low as a few nanometers that they could detect. They could hear the sound of a single bacterium. The extremely small oscillations are a result of the biological processes of the bacteria with main contribution from their flagella, that is tails on the cell surface that propel bacteria. These flagellar beads on graphene are at least 10 billion times smaller than a boxer's punch when reaching a punch bag. Yet these nanoscale beads can be converted to soundtracks and listened to. This research has enormous implications for the detection of antibiotic resistance. The experimental results were unequivocal. If the bacteria were resistant to the antibiotic, the oscillations just continued at the same level. When the bacteria were susceptible to the drug, vibrations decreased until 1 or 2 hours later, but then they were completely gone. Thanks to the high sensitivity of graphene drums, the phenomenon can be detected using just a single cell. For the future, the researchers aim at optimizing their single-cell graphene antibiotic sensitivity platform and validate it against a variety of pathogenic samples. Eventually, it can be used as an effective diagnostic toolkit for fast detection of antibiotic resistance in clinical practice. This would be an invaluable tool in the fight against antibiotic resistance, an ever-increasing threat to human health around the world.